The bone looks almost too small to carry a human story. It lies in the cold dark of a cave, sealed in mud, mineral dust, and the breath of thousands of winters. No name remains with it, no language, no memory of the person who once moved through the world with hunger, fear, warmth, and purpose. For most of history, this fragment would have been nearly silent. An archaeologist might measure it, compare its shape, place it beside stone tools or animal bones, and cautiously imagine a life around it. But the deeper past is often cruel with evidence. It breaks bodies apart, it scatters settlements beneath ice, sea, forest, desert, and time. Whole peoples vanish so thoroughly that they leave behind only a tooth, a finger bone, a stain in the ground, or nothing visible at all. Then, from inside the bone, something impossible begins to speak. Not a voice, not a memory in the way stories preserve kings or battles, but a chemical trace, a code written in the cells of someone who died before any empire, before any city, before writing, before history knew how to remember. Ancient DNA changes the atmosphere around the dead. A cave floor is no longer only a place where bones have lain undisturbed. It becomes an archive. A tooth is no longer only a tooth. It becomes a sealed message. A skeleton is no longer simply evidence of death. It becomes a map of ancestry, movement, kinship, disease, survival, and disappearance. This is one of the strangest turns in the study of the human past. For centuries, the deep story of humanity had to be reconstructed from what survived outside the bodystone blades: hearths, beads, burials, broken pottery, painted walls, footprints hardened in mud. These traces are powerful, but they are also incomplete. A stone tool can show skill, but not always who made it. A burial can show care, but not always whether the buried person belonged to the people who placed them there. A skull can suggest relationship, but bones can mislead when different populations adapt to similar climates or lifestyles. The human past was visible mostly as shadow and silhouette. Ancient DNA does not remove the mystery. In some ways, it deepens it, but it gives the shadows edges. Inside the cold-preserved bones of Ice Age hunters, inside the teeth of early farmers, inside the remains of people buried under plague cemeteries, scientists find patterns that no one expected. Populations once imagined as stable turn out to have been replaced, absorbed or transformed. Groups that archaeologists could barely see become visible as genetic ghosts. People thought to be separate become intertwined. People thought to be ancestors of everyone turn out to be ancestors of only some. And in the genomes of living humans, fragments of vanished relatives still persist, quiet inheritances from meetings that happened tens of thousands of years ago. Before we go deeper, take a moment to like the video and subscribe, if this is the kind of history and mystery you enjoy. The first surprise of ancient DNA is not simply what it reveals, but that it survives at all. DNA is fragile. After death, the body becomes a landscape of decay. Cells rupture, microbes invade, water seeps through bone, heat breaks chemical bonds, time cuts long genetic strands into smaller and smaller fragments, like a manuscript left in the rain and then burned at the edges. In warm, wet places, DNA can vanish quickly, in cold caves, dry deserts, permafrost, and protected teeth. It may endure far longer, though never untouched. Ancient DNA is usually damaged, contaminated, and mixed with the genetic material of soil organisms, animals, and the modern people who excavated or handled the remains. The work of recovering it is therefore part science, part forensic patience, and part ritual of caution. Researchers enter clean rooms wearing full-body suits, masks, gloves, and hoods, not to protect themselves from the ancient dead, but to protect the ancient dead from them. Modern human DNA is everywhere. It floats from skin, breath, hair, and dust. A single careless touch can overwhelm the faint signal from a person who died 40,000 years ago. Bone is drilled, powder is collected. Chemical processes separate tiny fragments of DNA from the mineral structure that protected them. Machines read these fragments, not as whole books, but as torn letters. Computers then attempt the reconstruction, comparing fragments, identifying damage patterns, filtering contamination, and assembling enough of the genome to see who this person was related to. There is something quietly astonishing about that process. The dead do not return as faces, not at first. They return as relationships. A child in an ancient grave becomes the daughter of two parents whose ancestry came from different worlds. A hunter buried with care becomes part of a lineage that no longer exists in pure form. A woman from a cave becomes evidence that a population crossed a continent earlier than anyone thought. A man beneath a burial mound becomes a clue to migrations that helped reshape languages and societies far beyond his own horizon. And the revelations do not always match the stories humans later told about themselves. Modern identities tend to imagine deep roots. Peoples picture themselves as rising from one place, one homeland, one unbroken line. Ancient DNA shows something far more restless. Again and again. The human past is made of movement, not one migration, but many. Not clean replacement or simple continuity, but layers arrivals, mixtures, collapses, survivals, expansions, vanishings. A landscape that appears timeless may have held several different human worlds, each leaving traces in the next. A valley may contain the genes of Ice Age foragers, incoming farmers, steppe herders, traders, captives, invaders, and neighbours who became kin. What survives in the record suggests that ancestry is rarely a straight line. It is a braided river, splitting and rejoining across thousands of years. Yet ancient DNA also exposes the limits of what can be known. A genome can show that two groups mixed, but not whether they met through marriage, alliance, hunger, slavery, adoption, raiding, refuge, or slow neighbourly exchange. It can show that a population declined, but not always whether drought, disease, violence, social breakdown, or simple absorption caused the disappearance. It can reveal kinship inside a grave, but not grief. It can trace a pathogen, but not the terror it caused as fever spread from house to house. The evidence remains incomplete. DNA is powerful, but it is not a complete resurrection of the past. It must be read beside archaeology, climate records, language, oral tradition, and the stubborn physical details of places where people lived and died. That balance matters because ancient DNA has become one of the most dramatic scientific tools ever applied to human history. It has solved mysteries, overturned assumptions, and opened others that are even stranger. It reveals that some of our ancestors were not quite who we imagined. It shows that modern humans met other kinds of humans, and that those meetings left biological consequences still present in people today. It shows that Europe, Asia, Africa, Oceania, and the Americas were not populated by simple waves of anonymous migration, but by real communities adapting to collapsing ice sheets, changing coastlines, spreading forests, growing deserts, new diseases, and each other. In the beginning, there was only the fragment of bone under ice, a tooth in a cave, a skeleton folded into the earth. Around it is silence. Above it, landscapes change beyond recognition. Glaciers retreat, seas rise, forests advance, rivers shift, languages are born and lost. The descendants of some people spread across the planet, while others disappear so completely that no story remembers them. But inside the small surviving pieces of the body, the past keeps one last form of memory. And when that memory is opened, it does not tell a simple tale of progress. It tells a stranger story that humanity is a survivor of meetings with the vanished, of disasters endured, of populations swallowed and reborn, of forgotten ancestors whose names are gone, but whose traces remain in blood, bone, and breath. The first people to rise from ancient DNA are not always the people archaeologists can see. They do not stand in painted caves or lie beneath grand tombs. They appear instead as absences that suddenly acquire shape, as shadows behind other shadows. For a long time, the human past was limited by the bones that happened to survive. If no skeleton was found, no population could be measured. If no skull emerged from a cave or riverbank, an entire people might remain unknowable. Archaeology could sense missing pieces, but it could not always name them. Ancient DNA changes that by revealing ancestors who left almost no visible remains at all. These are often called ghost populations, not because they are supernatural, but because their presence is inferred from traces inside other genomes. They are populations that contributed ancestry to later people, yet have not been directly sampled from ancient remains or have only been glimpsed through tiny fragments. The phrase sounds dramatic, but the idea is sober and mathematical. When researchers compare DNA from living and ancient humans, they sometimes find patterns that cannot be explained by known groups alone. A population carries genetic signals that seem to come from a source no one has found in the ground. Somewhere, at some time, there must have been another group. It lived, reproduced, mixed with neighbours, and vanished as a distinct people before leaving behind enough bones to be recognised. This is one of the quiet revolutions of the field. The past is no longer limited to what can be placed in a museum drawer. It can sometimes be reconstructed from what passed through people into their descendants. The idea unsettles older images of prehistory. It is tempting to picture ancient humanity as a set of clear branches, one group here, another there, each evolving separately until one replaces another. But ancient DNA reveals a landscape more tangled than a tree. It resembles a thicket. Populations split, drift apart, meet again, exchange genes, separate, shrink, expand, and sometimes disappear into the ancestry of others. Some vanish because they are overwhelmed by newcomers. Some blend gradually until their separate identity is no longer visible archaeologically. Some survive in remote regions for thousands of years, leaving genetic echoes far from where anyone expected them. One of the most surprising lessons comes from the fact that a ghost population can be real even before a single bone is found. In the genomes of some ancient and living people, scientists detect signals of archaic human ancestry that do not match Neanderthals or Denisovans neatly. This does not mean a hidden civilization or a lost kingdom in the popular sense. It means that the human family was more diverse than the fossil record alone shows. There may have been deeply separated populations of archaic humans in Africa and Eurasia that interacted with the ancestors of later humans, then disappeared as distinct groups. Scholars still debate how many such populations existed, where they lived, and how best to describe them, because genetic models can suggest possibilities without providing the full human context. The deeper one goes, the stranger the family album becomes. Africa, where Homo sapiens emerged, contains the greatest human genetic diversity on Earth. That diversity is not a footnote. It is the foundation of the entire story. For too long, simplified accounts treated Africa mainly as the place humans left. Ancient and modern DNA show, instead, that Africa itself held a vast, complex, shifting world of populations, some separated for immense stretches of time. Many of these groups left few ancient genomes because warm climates are harsh on DNA preservation. Heat and humidity destroy the fragile molecule quickly. As a result, the continent most central to human origins is also one of the hardest places for ancient DNA to speak clearly. What survives in the record suggests that many forgotten lineages remain hidden there, not because they were unimportant, but because chemistry has been unkind. A lesser-known detail is that sometimes the soil itself can reveal vanished humans. Researchers have learned to recover ancient DNA not only from bones and teeth, but from cave sediments. A cave floor can contain microscopic fragments shed from skin, blood, urine, feces, or decomposing bodies. No skeleton is needed, the ground becomes a genetic trap. In some sites, sediment DNA has shown the presence of Neanderthals, Denisovans, or modern humans even when identifiable bones are absent. This changes the meaning of an empty cave. A layer of dirt may contain the trace of a population passing through in a season no artifact can clearly assign. The human body leaves itself behind in ways no one could see until recently. Yet ghost populations must be handled with care. Genetic models depend on comparisons, assumptions, and available samples. If the reference populations are incomplete, the missing source may appear more mysterious than it really is. A ghost may later gain a body when a new ancient genome is found. A puzzling ancestry component may be reinterpreted as more samples arrive. No single explanation fully accounts for every strange pattern, and the field changes quickly. Ancient DNA is powerful precisely because it can test old ideas, but it also creates new uncertainties, especially when the dead are represented by fragments and probabilities rather than whole communities. Still, the implications are profound. To understand forgotten ancestors, one must accept that disappearance is not the same as extinction. A people can vanish as a separate group while still living on inside others. Their tools may be reclassified, their language lost, their burial customs forgotten, their faces unrecorded, but their ancestry may continue quietly for thousands of generations. They become invisible culturally and archaeologically, yet biologically present. This is one reason ancient DNA feels so intimate. It does not merely say that unknown people once existed. It says they helped make later humans possible. The ghost populations also remind us that prehistory was crowded, not necessarily crowded in the modern sense, with dense cities and borders, but crowded with difference. The world of ancient humans contained groups whose names are unrecoverable, whose territories shifted with animals, rivers, glaciers, and rains. They may have recognized each other by speech, ornaments, body markings, songs, hunting practices, marriage rules, or stories that no longer survive, to genetics. They appear as clusters and admixture events. To themselves, they were not data points. They were people with kin, enemies, obligations, fears, and remembered dead. This is where the mystery becomes larger than science alone. Ancient DNA can reveal that a population existed, but it cannot fully tell us what it meant to belong to that population. It cannot recover a lullaby, a taboo, a joke told beside a fire, or the name of a dangerous mountain pass. The genetic ghost is real, but incomplete. It is a door opened into a room where most of the objects have been removed. And yet, even that partial opening transforms the human story. The forgotten ancestors are no longer merely those who left monuments or myths. They include the ones who left almost nothing except a faint signal in later blood. They are the missing presences behind the known dead, the unseen contributors to the living world. Once ancient DNA reveals them, history becomes less like a procession of famous cultures and more like a vast buried network, full of vanished branches feeding roots that still reach upward. From these ghostly traces, the story moves toward a more recognizable vanished relative, one whose bones were found before the code inside them could be understood. For more than a century, Neanderthals stood beside modern humans as a separate, heavy-browed mystery. Ancient DNA would eventually show that they were not simply neighbours in the Ice Age. The Neanderthal story begins in a valley, with bones that look too human to dismiss and too strange to comfortably accept. When the first recognised Neanderthal remains emerged in the 19th century, they entered a world that was only beginning to understand deep time. The idea that humans had ancient relatives was unsettling enough. The idea that another kind of human had once lived across Europe and Western Asia, endured Ice Age cold, made tools, buried some of their dead, survived for hundreds of thousands of years, and then disappeared, was almost too large to fit inside the imagination of the age. For decades, Neanderthals were made into whatever modern people needed them to be: brutes, failures, missing links, noble survivors, evolutionary warnings. Their bones became a mirror, and the reflection often said more about the living than about the dead. Ancient DNA changed that mirror into something sharper. When scientists finally recovered Neanderthal DNA, the question was no longer only whether they looked like us, thought like us, or competed with us. The question became whether they had become part of us. For a long time, many researchers suspected that modern humans spreading out of Africa replaced Neanderthals with little or no interbreeding. It was a clean story, and clean stories are often attractive. One population expands, another disappears, the world moves on. But the genome is rarely so tidy. When the first high-quality comparisons were made, they revealed that many living people outside Africa carry a small percentage of Neanderthal ancestry. The number is usually modest, often around 1 to 2%, but its meaning is enormous. It means that modern humans and Neanderthals met not only as rivals or strangers, but as people close enough biologically and socially to have children together. Those children lived. Some had children of their own. Across tens of thousands of years, most individual Neanderthal ancestors vanished into a vast human genealogy, but fragments of their DNA remained. This inheritance is not spread evenly. Different living populations carry different pieces. No living person has a complete Neanderthal genome, but together, modern humans preserve much of the Neanderthal genetic legacy in scattered fragments. It is as if a shattered manuscript has been distributed across millions of bodies. One person carries a paragraph, another carries a torn sentence, another carries only a few letters, together. They reveal that the vanished people of Ice Age Eurasia did not disappear in a simple biological sense. Their distinct populations ended, but parts of them crossed the threshold into us. The details are stranger still. Neanderthal DNA appears to influence traits connected to skin, hair, immunity, metabolism, and responses to infection. Some inherited variants may have helped early modern humans adapt to Eurasian environments, especially after leaving Africa and encountering new climates, new pathogens, and unfamiliar seasonal pressures. An immune-related gene acquired from archaic humans could be useful in one setting and harmful in another. This is one of the subtle truths ancient DNA reveals. Inheritance is not a moral gift or curse. It is a historical residue. A genetic variant shaped by Ice Age survival may carry different consequences in a modern world of cities, changed diets, longer lifespans, and different diseases. There is a lesser-known and haunting clue in the pattern of Neanderthal ancestry itself. Some regions of the modern human genome contain very little Neanderthal DNA, less than random mixing would predict. This suggests that some Neanderthal variants were gradually removed by natural selection, perhaps because they were harmful in modern human genetic backgrounds, or affected fertility. The evidence remains incomplete, and scholars still debate the strength and timing of these processes. But the pattern hints that interbreeding was not a simple blending of equals at the genetic level. Some inherited fragments endured, others faded. The children of these encounters carried both possibility and burden. The human side of those encounters remains mostly beyond reach. Ancient DNA can show that interbreeding happened, and sometimes roughly when. It cannot tell us whether a particular meeting occurred through cooperation, captivity, attraction, alliance, violence, or the slow familiarity of neighbouring groups using the same values over generations. Archaeology shows that Neanderthals were capable hunters and toolmakers, with regional traditions and a deep knowledge of difficult landscapes. They lived through glacial cycles that transformed Europe into a shifting mosaic of steppe, forest, tundra, and refuge zones. They followed animals, used fire, crafted stone tools with skill, and occupied caves whose entrances overlooked vanished plains. Some sites suggest symbolic behaviour, though the interpretation of certain ornaments, pigments, and burial practices remains debated. What survives in the record suggests a human world, not a caricature. Their disappearance was not a single event. Neanderthals did not vanish everywhere on the same day, under one catastrophe, in one final dramatic scene. The decline likely unfolded unevenly across regions. Climate instability may have stressed small populations. Modern humans arrived with different technologies, wider exchange networks, or more flexible social structures. Competition for prey and territory may have mattered. Disease may have passed between groups. Low population density may have made Neanderthals vulnerable to chance losses, inbreeding, or local collapse. No single explanation fully accounts for this. The end of the Neanderthals was probably not one clean cause, but a convergence of pressures acting on people already living close to the edge. Ancient DNA has made that edge visible. Neanderthal genomes show signs of small population sizes and long-term isolation in some groups. In certain late Neanderthal remains, genetic evidence points toward close parental relatedness, suggesting communities that may have been tiny and fragile. Imagine a group in a cold valley where every adult matters, where knowledge of stone, fire, roots, and animals is held by few people, where a failed hunt, a harsh winter, or the loss of several children can change the future of an entire lineage. This is not the image of a stupid species outcompeted by brilliance. It is the image of vulnerable humanity in a dangerous world. And yet, vulnerability did not mean simplicity. Neanderthals endured far longer than many later civilizations. They lived in Europe and Western Asia across enormous stretches of time, through climates that would erase most human settlements from memory. The Roman Empire lasted centuries. Neanderthals lasted hundreds of thousands of years. Measured that way, their story does not look like failure. It looks like astonishing persistence, followed by disappearance under conditions we still do not fully understand. The deeper surprise is that their disappearance did not remove them from the future. A person walking through a modern city may carry Neanderthal variants inherited from encounters that happened when mammoths still moved across cold grasslands and glaciers held mountains in white silence. These fragments do not make anyone less modern. They make modern humanity more complicated. They show that Homo sapiens did not walk into Eurasia as a sealed, untouched lineage. We entered a world already inhabited by other humans, and we became, in part, the result of meeting them. This changes the emotional weight of the Neanderthal bones. A skull in a museum is not only the remnant of an extinct relative, it is also evidence of kinship. The heavy brow, the strong body, the broad nose shaped for cold air, the hands that made tools and carried meat and perhaps touched the faces of children. belong to a people who are gone and not entirely gone. They survive in fragments, not as a living culture, not as a separate humanity, but as a faint biological afterimage inside us. Beyond them, another vanished human waits in even deeper obscurity. Unlike Neanderthals, this group did not first appear through a famous skull or a nearly complete skeleton. It emerged from a few fragments in a Siberian cave, from DNA before anatomy, from a human presence with almost no face. The genome spoke first, and only afterward did the world learn the name Denisovan. The cave sits in the Altai Mountains, where Siberia gathers itself into ridges, forests, cold rivers, and long seasons of snow. From the outside, Denisova Cave does not look like a doorway into a missing branch of humanity. It looks like a shelter, a place where animals, wind, and people could escape the open landscape. But the floor of a cave is sometimes a slow machine for preserving time. Layer after layer settles. Ash from fires darkens the sediment. Stone tools fall and are covered. Animal bones are broken, gnawed, buried, and forgotten. A fragment of a bracelet, a bead, a tooth, a splinter of bone, each object enters the ground with no idea that thousands of years later it may force the human story to be rewritten. The Denisovans did not first arrive as a skull. They did not appear as a full skeleton standing beside Neanderthals in the museum imagination. They emerged from a finger bone so small it could almost be overlooked, a tiny piece of a child's hand found in Denisova Cave. At first, the bone did not announce a new kind of human. It was simply ancient, human-like, and promising enough for genetic analysis. Then the DNA inside it revealed something no one expected. It was not Neanderthal. It was not modern human. It belonged to a population related to Neanderthals, but distinct from them, a sister lineage that had split away deep in the past. A vanished human group had been discovered almost without a body. This is why Denisovans feel so unsettling. Neanderthals have faces in the public mind. Their skulls are heavy and recognisable. Their skeletons have weight. Denisovans, for a long time, were mostly genetic presence. Scientists knew them from code before they knew their shape, a few teeth, a jawbone from the Tibetan plateau later linked to them, small bone fragments, and traces in sediment have slowly added substance, but they remain far less visible than Neanderthals. It is possible to know that they existed across vast regions of Asia, and yet not be able to describe with confidence exactly what most Denisovans looked like. The evidence remains incomplete, and that incompleteness is part of the fascination. The genetic evidence, however, is extraordinary. Denisovan ancestry survives today in several living populations, especially among people in parts of Oceania, including Papua New Guinea and Aboriginal Australian populations, and in lower levels across parts of Asia. This creates an immediate geographic puzzle. The first Denisovan remains were found in Siberia, but some of the strongest Denisovan genetic signals appear far to the south and east, across island worlds and tropical regions where ancient DNA rarely survives. The implication is that Denisovans, or closely related Denisovan populations, were not confined to one cold mountain cave. They were likely spread across a huge and varied range, from northern Asia to regions much closer to Southeast Asia, and perhaps beyond. That means a tiny fingerbone in Siberia may represent only the northern edge of a much larger vanished world. The word Denisovan can make this world sound unified, as if there was one people with one culture, one territory, one way of life. Ancient DNA suggests something more complex. Denisovan ancestry in living humans appears to come from deeply separated Denisovan-related groups, not just one population. Some genetic studies indicate that modern humans may have interbred with Denisovan groups more than once, in different places and at different times. Scholars still debate the exact number of encounters and the geography behind them, but the broad picture is clear enough to be remarkable, as modern humans moved through Asia and into Sahul, the ancient landmass that joined Australia and New Guinea during lower sea levels. They encountered other humans whose histories had already been unfolding for hundreds of thousands of years. One of the most striking Denisovan inheritances concerns altitude. Some Tibetan populations carry a version of a gene associated with adaptation to high-altitude life, often linked to Denisovan ancestry. This variant helps the body cope with low oxygen environments in ways that appear beneficial on the Tibetan plateau. It's a powerful example of how a vanished population can shape the survival of later people in a specific landscape, a genetic fragment from an archaic human group. Preserved through interbreeding may have helped modern humans live where thin air makes ordinary biology struggle. The story is not romantic in a simple way. It is evolutionary and accidental, shaped by survival rather than intention. But it gives the Denisovan legacy a strained continuity. People with almost no surviving bones may have helped others thrive on the roof of the world. Still, the Denisovan world itself remains dim. The artifacts in Denisova Cave include sophisticated objects, but assigning them to a particular human group is difficult because the cave was used at different times by Denisovans, Neanderthals, and modern humans. A polished stone bracelet and other ornaments from the site have attracted attention, but scholars remain cautious about linking every object to Denisovan specifically, in a cave shared across millennia by multiple human groups. Culture does not always attach neatly to DNA. A tool layer may contain traces of one population, while a nearby object could belong to another. The ground keeps secrets even when it preserves evidence. Perhaps the most haunting discovery from Denisova Cave is not just that Denisovans existed, but that they met Neanderthals too. One bone fragment belonged to a girl whose mother was Neanderthal, and whose father was Denisovan. She has sometimes been described in scientific shorthand as a first-generation hybrid, but that phrase is too cold for what it implies. She was a child of two vanished human lineages, born into a world where different kinds of humans did not merely occupy separate territories on a map. They encountered one another closely enough to form families. Her existence suggests that these meetings may not have been rare accidents, even if the fossil record preserves only a few of them. The child's bone is a small object, but it breaks open a large idea. The Ice Age world was not divided cleanly between modern humans, Neanderthals, and Denisovans like separate nations with fixed borders. It was a shifting mosaic of populations, some isolated, some overlapping, some meeting in caves, river valleys, mountain corridors, and vanished plains. The categories we use today are necessary, but ancient life was probably messier. A person living then may not have thought in species terms at all. They would have recognised familiar people and strangers, possible mates and dangerous outsiders, neighbouring bands, different speech, different customs, different bodies perhaps, but not the tidy labels placed on them tens of thousands of years later. Denisovans also reveal the bias of preservation. If the first tiny bone had not been analysed, if ancient DNA technology had arrived later, if the cave had been warmer or wetter, this entire lineage might still be almost unknown. That should make us cautious. If one cave can reveal a missing humanity, how many other human groups have left no recoverable DNA, no recognised fossils, no name? The Denisovans are not merely an answer. They're a warning about the poverty of the surviving record. They show that the past may contain major populations hidden behind conditions of preservation, excavation, and chance. And so, the forgotten ancestors become less like a small cast of known characters, and more like a crowded landscape glimpsed through fog. Neanderthals are there, close enough to leave traces in many living people. Denisovans are there too, their outlines broader than their bones. Ghost populations gather behind them, modern humans move among them, not as solitary conquerors of an empty planet, but as newcomers entering older human worlds. The meetings are intimate, dangerous, fertile, and often invisible except in the genome. From caves and fragments, the story begins to stretch outward across continents. DNA does not stay in one place. It follows bodies across ice margins, grasslands, coastlines, deserts, and drowned plains. To understand what ancient DNA reveals next, the eye has to move from the bone in the cave to the landscapes those bones once crossed, many of which no longer exist in any recognisable form. The map of the Ice Age is not the map people know today. Coastlines sit in the wrong places, seas withdraw from shallow shelves, islands become hills on open plains, rivers run through lands now drowned beneath grey water, forests shrink, grasslands widen, deserts pulse between harshness and unexpected green, and glaciers press down on the northern world with a weight that bends the crust beneath them. To follow ancient DNA across this landscape, the mind has to let go of familiar borders. There is no English Channel in the way later history knows it. There is Doggerland, a low plain between Britain and Europe, crossed by animals and people before the sea takes it. There is Beringia, not merely a land bridge, but a vast cold region between Siberia and Alaska, with its own ecology and human possibilities. There is Sahel, where Australia, New Guinea, and Tasmania form one greater landmass during lower sea levels. The world of our ancestors is not simply older. It is physically different. Ancient DNA turns these lost landscapes into roots. A genome taken from a burial, a cave, or a fragment of bone can reveal movement that no road records, no inscription names, and no later tradition remembers. It can show that people travelled farther, mixed more often, or endured isolation longer than archaeologists once believed. Migration no longer appears as a single arrow drawn across a textbook map. It becomes a long sequence of advances, pauses, retreats, and encounters, shaped by climate, hunger, curiosity, social ties, and the slow pressure of survival. The first great movement out of Africa by the ancestors of most living non-African people was not a simple march into an empty world. Modern humans entered Eurasia, where Neanderthals already lived in many regions and Denisovan-related groups occupied parts of Asia. Earlier modern human expansions may have occurred and left limited genetic legacy, while later expansions became ancestral to many people alive today. The evidence remains incomplete, especially because ancient DNA from warm early sites is so difficult to recover. But what survives suggests a story of repeated movement rather than one heroic departure. Humans left, met barriers, found corridors, disappeared from some regions, expanded into others, and changed as they went. One of the most important forces behind these movements was climate instability. Ice Age climate did not simply mean cold, it meant fluctuation. Environments shifted over centuries and sometimes far faster. Grasslands opened and closed. Herd animals changed their ranges. Rivers altered courses. Lakes expanded and dried. A valley that supported hunters for generations might become less reliable, while another region opened as ice retreated or rains returned. Ancient people were not passive victims of these changes. They knew landscapes intimately, but their knowledge had to adapt to a world that kept remaking itself. Genomes carry the memory of those pressures. When a population becomes isolated, its DNA begins to show the effects of small numbers and limited mates. When groups meet after long separation, their descendants carry mixed ancestry. When a population expands rapidly, genetic patterns may preserve the signal of that growth. These signals do not show the creak of snow underfoot or the smell of wet hides drying near a fire, but they reveal the larger rhythm of human movement through changing Earth. A surprising detail lies beneath the sea. Many of the places Ice Age people used most heavily may now be underwater. Coastal routes were likely important for early migrations, because shorelines offered food, travel corridors, and ecological variety. But lower Ice Age sea levels placed those ancient coasts far beyond modern beaches. When the ice melted and seas rose, campsites, graves, hearths, and paths were submerged. This means the archaeological record is biased toward inland places and caves that happen to survive above water. Ancient DNA sometimes helps fill the absence, showing movement along routes whose physical evidence may lie drowned on continental shelves. The silence of the coast is not proof that people were absent. It may only mean that the sea has taken the archive. In Eurasia, ancient genomes reveal population turnovers and long-distance connections among hunter-gatherers after the last glacial maximum. Groups that survived in refuges during the coldest periods later expanded into newly habitable lands. Europe, for example, was not continuously occupied in the same way across all regions. Ice sheets and brutal climates pushed human groups southward or into pockets where survival was possible. As conditions warmed, people moved again into forests. River systems and plains, the genetic landscape changed with them. Some lineages flourished, others faded, and some reappeared in unexpected mixtures. Farther east, the story becomes even more complex. Ancient DNA from Siberia, East Asia, and Central Asia shows that populations were repeatedly reshaped by migrations across enormous distances. The steppe, forest, tundra, and mountain corridors did not act as permanent walls. They were difficult, but not empty. People moved through them, sometimes slowly, sometimes across generations, carrying technologies, ancestry, and perhaps stories of animals, spirits, and landmarks now lost. The genetic connections between ancient Siberians, East Asians, Native Americans, and other groups reveal that Northern Asia was not a remote margin. It was one of the great theatres of human prehistory. Bringer is especially important because it was not simply a bridge crossed once and forgotten. During lower sea levels, it formed a broad region linking Northeast Asia and Northwest North America. Some models suggest that ancestors of Native Americans may have spent time isolated in or around Beringia before spreading south into the Americas. Ancient DNA supports a complex picture of related but distinct populations, splits, and later movements. Scholars still debate the exact routes, timing, a number of movements into the Americas, and the evidence is sharpened or complicated by every new genome. But the genetic record makes clear that the peopling of the Americas was not a crude tale of a single band walking over a narrow strip of land. It was a deep human process unfolding across a vanished northern world. In Southeast Asia and Oceania, ancient DNA faces the problem of heat and humidity, but living genomes preserve signals of very old journeys. The movement into Sahul required sea crossings, because even at low sea levels, water barriers remained. This means that tens of thousands of years ago, modern humans reached lands that demanded planning, maritime skill, or at least repeated successful crossings through island chains. The people who entered Sahul encountered unfamiliar animals, climates, and landscapes, and they also carried Denisovan ancestry that points back to meeting somewhere along the route. The physical remains are sparse compared with the magnitude of the journey. The genetic record helps reveal how extraordinary it was. But migration is not only expansion. Sometimes it is disappearance. Ancient DNA shows that groups once present in a region may contribute little or nothing to later inhabitants. This can happen through population replacement, extreme bottlenecks, absorption, or simply the uneven survival of lineages. It is easy to imagine migration as progress. As humanity is spreading confidently outward, the genetic evidence is more severe. For every expansion that becomes ancestral to millions, other paths end. A group crosses a plain, survives for generations, adapts to a valley, then leaves no clear descendants. Another population persists through a glacial crisis, but is later absorbed by newcomers. Some lineages become central to later history, others become faint traces or vanish entirely. This is where ancient DNA brings a particular kind of gravity. It shows movement, but also fragility. Human groups can cross continents, endure cold, navigate seas, and adapt to new worlds, yet still disappear as recognizable populations. Their success in one age does not guarantee survival in another. The land changes, animals move, neighbours arrive, children are born into different networks than their grandparents knew. Identity dissolves into ancestry. The forgotten ancestors were not only buried in caves and graves, many were buried beneath geography itself. Their valleys are flooded, their hunting grounds lie under farms, cities, or seabeds. Their trails have become straits. Their coastlines are gone, but ancient DNA allows their movements to rise through the altered world, revealing a planet that humans crossed while it was still becoming the one we recognise. The story now narrows toward Europe, not because Europe contains the oldest human story, but because its cold caves, dense archaeology, and well-preserved bones have made it one of the clearest laboratories for seeing how dramatically ancient. Europe before civilization is often imagined through a few familiar images: hunters in fur moving across frozen plains, torchlight flickering over painted animals in caves, mammoths in the distance, and small bands surviving at the edge of glaciers. Those images are not entirely wrong, but they are far too still. Ancient DNA reveals a continent in motion, repeatedly emptied, refilled, divided, and transformed long before farming cities or written memory. The people of Ice Age Europe were not one unchanging population waiting for agriculture to arrive. They were many populations, sometimes deeply related, sometimes surprisingly distinct, shaped by cold, refuge, extinction, return, and the unstable generosity of the land. During the harshest phases of the last Ice Age, much of northern Europe became difficult or impossible for humans to occupy for long. Ice sheets covered huge areas. Tundra and steppe stretched across regions that would later become forest, field, and city. Survival depended on mobility, knowledge of animals, and access to places where climate allowed enough food to be found. Southern Europe, including parts of Iberia, Italy, the Balkans, and areas around the Black Sea, may have served as refugees at different times, though the exact population histories are still debated. What survives in the genetic record suggests that people retreated, endured in pockets, and later expanded again when warming opened the continent. This movement left marks in ancient genomes. Some hunter-gatherer lineages appear, disappear, and reappear across thousands of years. At times, genetic ancestry associated with earlier Ice Age Europeans becomes rare or vanishes from certain regions, replaced by people with different ancestry profiles. Later, as ice retreats and forests spread, new hunter-gatherer groups occupy landscapes that would have been unrecognizable to their predecessors. Europe's deep past is not a smooth ancestral chain. It is a sequence of broken continuities. One of the surprising details revealed by ancient DNA is how physically different some Mesolithic Europeans may have looked from many later Europeans. Certain Western hunter-gatherer genomes carry genetic variants associated with dark skin pigmentation and blue eyes, a combination that does not fit many modern assumptions about ancient Europe. This does not mean every hunter-gatherer looked the same, and translating genes into appearance is not always simple. Still, the finding shows how misleading it can be to project present-day populations backward unchanged. The faces of prehistoric Europe were not just earlier versions of modern national portraits. They belonged to worlds that no longer exist. The older archaeological record had already shown cultural richness. Cave paintings, carved figurines, decorated tools, personal ornaments, and careful burials all point to symbolic lives of great complexity. But ancient DNA adds another layer. The people who made these objects were connected and separated in ways that artifacts alone cannot always reveal. Similar tools do not always mean the same ancestry. Different tool traditions do not always mean different biological populations. Culture can spread without mass migration, and people can move without immediately changing every object they use. This is one of the great lessons genetics forces into archaeology. Pots, points, and ornaments matter deeply, but they do not speak for blood by themselves. In some cases, ancient DNA confirms dramatic population shifts that archaeologists had suspected only dimly. In others, it complicates the meaning of cultural change. A new style of tool may appear in a region, but the people using it might be local descendants adopting new techniques elsewhere. A new burial custom may arrive with migrants. The evidence has to be read carefully, site by site, period by period. Scholars still debate how to connect genetic ancestry with archaeological cultures. Because culture is not a gene and ancestry is not identity. A person may inherit DNA from one group, tools from another, and burial rites from a third. The end of the Ice Age does not bring immediate simplicity. As temperatures rise, forests spread over landscapes once dominated by open steppe. Large cold-adapted animals decline or move north. Human lifeways change. Hunting strategies adapt to deer, boar, fish, birds, nuts, and seasonal plant resources. Rivers and coasts become increasingly important. The sea rises, swallowing lowlands and cutting Britain from the continent. Doggerland, once a broad plain, slowly becomes marsh, island. And then memory beneath the North Sea. The people living through this transformation do not know they are witnessing the end of a world. They only know that familiar hunting grounds become wetter, that paths change, that animals shift, and that the sea keeps coming. Ancient DNA from this period reveals hunter-gatherer groups with distinct regional ancestries. Western hunter-gatherers, Eastern hunter-gatherers, Scandinavian hunter-gatherers, and other labels used by researchers are not tribes in the way that people would have named themselves. They are scientific categories, useful but limited. Behind each label were communities with their own territories, marriages, seasonal movements, and memories. The genetic distinctions show that Europe's foragers were not one homogeneous population. Even among people living by hunting, gathering, and fishing, geography and time created deep differences. The human consequences of those differences are easy to overlook. A river valley might connect communities through exchange and marriage. A mountain range might slow movement for generations. A warming climate might open a corridor, allowing groups that had been separate to meet. A coast rich in fish and shellfish might support denser settlement than an inland forest. Over time, these choices and constraints shaped ancestry. DNA records them as percentages and components. But in life, they were decisions about where to camp, whom to marry, which neighbours to trust, and when to move children away from danger. There is also violence in the record, though not everywhere and not always in the forms later history makes familiar. Some prehistoric European sites show traumatic injuries, massacres, or deaths that suggest conflict. Others show care, continuity, and long use of place. Ancient DNA can identify kin groups buried together, outsiders buried among locals, or individuals whose ancestry suggests movement across regions. It can reveal that a grave once interpreted as simply symbolic also contains a family relationship, or that people buried together were not closely related at all. Yet it cannot explain every wound. A skull fracture may be battle, ambush, ritual killing, accident, or something stranger to us. The evidence remains incomplete. By the time farming begins moving into Europe, the continent is already filled with deep histories. The first farmers do not enter an empty land. They encounter communities whose ancestors survived the Ice Age, watched forests replace tundra, adapted to new coasts, and maintained their own ways of life for thousands of years. These hunter-gatherers are sometimes absorbed, sometimes displaced, sometimes live alongside farmers for long periods. Their ancestry persists unevenly in later Europeans. Stronger in some regions, weaker in others, braided into new populations that neither side could have predicted. Ancient DNA makes this encounter visible with unusual force. It shows that the arrival of agriculture was not merely the spread of an idea from one village to another. In many places, it involved the movement of people. The continent that had already been remade by ice, warming, and sea level rise was about to be transformed again by seeds, animals, houses, and a new relationship with land. The foragers of Europe had endured deep time, but endurance does not make a world immune to change. Across the southeast, new families were already moving with wheat, barley, sheep, goats, cattle, and a way of life that would alter ancestry, diet, disease, labour, and the meaning of home. The first farmers arrive in Europe not as an abstract revolution, but as families carrying seeds, animals, habits, and a different rhythm of time. They come from the southeast, ultimately connected to farming populations of Anatolia and the Near East, where agriculture had already changed the relationship between people and the earth. Their world is built around planted fields, stored grain, managed herds, houses that remain in place, and landscapes altered by repeated human intention. They bring wheat and barley into lands where wild foods had long shaped the seasonal movements of hunter-gatherers. They bring sheep, goats, cattle, and pigs into forests and river valleys where earlier communities followed deer, boar, fish, birds, and nuts. They bring pottery, polished stone axes, new burial customs, and a way of life that seems stable from a distance, though it is vulnerable in its own ways. Ancient DNA shows that this change is not only cultural. In much of Europe, farming spreads with people. The early farmers are genetically distinct from many of the hunter-gatherers they encounter. They do not simply teach local foragers how to farm and vanish into them. They move in large enough numbers to alter the ancestry of the continent. This was one of the great surprises when ancient genomes began to accumulate. Older debates had often asked whether farming spread mainly by ideas or by migration. The answer, at least in many regions, is that people moved with the idea. Fields were not only copied, they were carried. But the story is not one of total replacement. It is more human and more uneven. In some places, farmers and hunter-gatherers seem to live near one another for centuries. In others, local forager ancestry enters farming communities gradually. Some early farming groups show little hunter-gatherer ancestry at first, then more in later generations, suggesting contact, intermarriage, absorption, or shifting social relations over time. There are places where the two worlds may have traded. Watched one another, competed for land, avoided one another, or formed alliances. A polished axe could move without a marriage. A person could marry across a boundary without transforming an entire economy. The evidence remains incomplete, but it does not support a simple picture of farmers sweeping across Europe like a single wave over empty sand. The landscape itself becomes part of the encounter. Farming requires different commitments. A planted field is a promise made to a place. It demands return, it demands protection, labour, and patience. A herd creates wealth that can walk away, die of disease, be stolen, or multiply. Stored grain offers security, but also attracts rodents, insects, raiders, and rot. Settled life can support larger communities, but it can also concentrate waste and disease to hunter-gatherers. The farmer's life may not have looked obviously superior. It was harder in some ways, narrower in diet at times, and tied to cycles of work that wild food economies did not share. Agriculture was not simply progress arriving with a basket of grain. It was a bargain with consequences. Ancient bones preserve some of those consequences. In early farming populations, archaeologists often find signs of changing diets, repetitive labour, disease burdens, and sometimes reduced stature or health stresses compared with some foraging groups. These patterns vary by region and period, and scholars still debate how to weigh them, but they remind us that farming did not immediately make life easier. It made life different. It allowed population growth, surplus, settlement, and eventually the foundations for towns, hierarchies, and states. But it also created new vulnerabilities: crop failure, dependency on a few foods, animal-borne diseases, property tensions, and conflict over land. A lesser-known detail is that milk was part of this story before most adults could easily digest it. Early farmers kept cattle, sheep, and goats, and dairy residues appear in pottery in some regions. Yet the genetic variant that allows many adults in Europe to digest lactose efficiently was not immediately common among the first farmers. It rose later, under conditions still debated. This means that ancient people may have processed milk into cheese, yogurt-like foods, or other fermented products that reduced lactose, long before adult milk drinking became biologically easy for many populations. The familiar modern image of pastoral Europeans drinking fresh milk is not a timeless inheritance. It is the result of a long genetic and cultural history. The meeting between farmers and foragers could also be tense. Some Neolithic sites show violence bodies in pits, skull injuries, mass graves, settlements with defensive features. It would be careless to imagine the entire farming expansion as peaceful diffusion, but equally careless to imagine constant war. The archaeological record suggests variation. Some communities were destroyed violently, others endured for generations. Some landscapes show coexistence, exchange, and mixed ancestry. No single explanation fully accounts for this transformation across a whole continent. Farming spread through migration, imitation, demographic growth, intermarriage, pressure, opportunity, and sometimes force. DNA gives this vast process a family-level intimacy. It can show that individuals buried in the same cemetery had different ancestry profiles, implying movement and mixture within communities. It can reveal that a person raised in one genetic tradition was buried according to another cultural practice. It can expose kinship patterns, showing whether certain tombs held biological relatives or social groupings that cut across bloodlines. In some monumental graves, related men may appear in privileged positions, hinting at emerging patrilineal structures and inherited status. In other places, burial tells a more complicated story. The genome does not replace archaeology, but it sharpens the questions: who belonged, who married in, who was excluded, who inherited, who moved. The arrival of farming also changes time itself. Hunter-gatherer time is often deeply seasonal, moving with migrations, ripening plants, fish runs, animal behaviour, and weather. Farming time is still seasonal, but it is tied to fields that must be cleared, planted, guarded, harvested, stored, and planted again. Memory attaches to houses and plots. Ancestors may become linked to land in new ways. Boundaries matter differently when labour has been invested in soil. Children inherit not just knowledge of roots and resources, but claims, obligations, and perhaps inequality. Ancient DNA cannot read a boundary marker made of memory, but it can show the population growth and social restructuring that follow when land becomes home in this new sense. Still, the hunter-gatherers do not simply vanish. Their ancestry persists, sometimes faintly, sometimes strongly, woven into later farming communities. In parts of Europe, forager ancestry increases after the earliest phases of farming, suggesting that local people were gradually absorbed or that mixed communities became more common. This genetic survival matters because archaeological narratives often unintentionally favour the visible newcomers. Farmers leave houses, pottery, fields, cemeteries, and dense settlements. Foragers often leave lighter traces. Without DNA, it is easier to mistake visibility for victory. Ancient DNA reveals that some of the older people of Europe remained inside the New World, even when their lifeways faded. The result is not simply Europe becoming agricultural, it is Europe becoming layered again. Ice Age hunter-gatherer ancestries, Anatolian farmer ancestries, local mixtures, regional differences, and new social systems combine into a continent more complex than before. Farming lays the groundwork for later population growth, trade networks, monuments, and conflict. It also sets the stage for another transformation, one that arrives from the grasslands to the east with animals, wheels, wagons, and a different kind of mobility. The farmers had changed Europe by binding people more tightly to land. The next great movement would come from people whose power grew from moving across open horizons. Beyond the settled villages and fields of Neolithic Europe, beyond the forest edges and river valleys where farming communities clear land and build houses, the grasslands stretch eastward in a scale that changes the imagination. The steppe is not empty. It only appears empty to people who do not know how to read it. It is a world of distance, wind, grass, animals, seasonal movement, burial mounds, and paths that do not need paving because hooves and wheels can make their own roads. Here, in the vast spaces north of the Black Sea and the Caspian Sea, ancient DNA reveals another transformation-gathering force long before anyone writes its history down. The people often associated with this change are linked to steppe pastoralist cultures, especially the Yamnaya horizon and related groups. They are not the first herders, nor are they a single tribe moving in perfect unity. They belong to a broader world of mobile pastoralism, where cattle, sheep, goats, and eventually horses shape wealth, movement, and power. Their ancestry itself is mixed. Formed from earlier Eastern European hunter-gatherers and populations connected to the Caucasus and nearby regions. Already, before they move westward and eastward, they are the product of older meetings. Ancient DNA rarely finds pure origins. It finds mixtures that later become origins for someone else. When steppe ancestry appears in ancient European genomes during the 3rd millennium BCE, the scale is startling. In many regions, especially northern and central Europe, people associated with the corded-ware complex carry a large proportion of steppe-related ancestry. This is not a faint trace. It suggests substantial migration and demographic change. The earlier farming populations are not erased everywhere, and regional patterns vary. But the genetic shift is one of the most dramatic detected in prehistoric Europe. A continent already transformed by farmers is transformed again by people whose lifeways are tied to herds, wagons, and open country. The horse stands near the centre of the popular image, though scholars still debate the exact timing and nature of early horse domestication, riding, and chariot warfare. It is tempting to imagine mounted warriors sweeping across Europe in a single overwhelming surge, but the evidence is more complicated. Early steppe mobility likely involved wagons drawn by cattle, as well as horses used in different ways over time. The full military revolution of horse-drawn chariots comes later in other contexts. Still, horses mattered deeply, even before the most dramatic forms of mounted warfare. The ability to manage animals across large spaces changed what distance meant. Herds could be moved, families could travel, goods, ideas, and people could cross landscapes that would slow settled farmers. A lesser-known but important detail is the role of wagons. The steppe was not only a mounted world, it was also a wheeled one. Wagons allowed households, supplies, and perhaps social worlds to move with herds. The mobility of pastoralists did not mean rootlessness. It meant a different relationship to place, one based on routes, seasonal ranges, water sources, and ancestral burial landscapes rather than fixed fields alone. A village farmer's world might be anchored by house and soil. A steppe herder's world might be anchored by movement itself, by the memory of where grass returns and where the dead lie beneath mounds. Those mounds, or kurgans, give the steppe a monumental presence. Burials beneath earthen barrows appear across wide regions, though their meanings vary through time and culture. Some graves contain bodies placed with ochre, tools, ornaments, weapons, carts, or animal remains. They do not speak in sentences, but they suggest societies where status, ancestry, and display mattered. Ancient DNA has sometimes shown strong male line patterns in steppe-associated expansions, raising the possibility that certain paternal lineages spread with unusual success. Scholars still debate the social mechanisms behind this elite dominance: clan expansion, warfare, marriage patterns, demographic advantage, or combinations of these. The genetic signal is clear in places, the lived social explanation remains harder to recover. The consequences reach far beyond bones. Steppe migrations are deeply entangled with debates about the spread of Indo-European languages, the enormous language family that includes many languages of Europe and parts of Asia. Ancient DNA cannot identify a spoken language directly. A genome has no grammar, but it can reveal migrations large enough to plausibly carry languages across regions. Many researchers see steppe expansions as central to the spread of at least some Indo-European branches, though the full story remains debated, especially for the earlier stages and for regions outside Europe. It is one of the most powerful examples of genetics entering an argument once dominated by linguistics and archaeology, not to settle every question but to reshape what explanations are possible. The human reality behind such language shifts must have been uneven. A language can spread through families, conquest, trade, prestige, intermarriage, or the slow adoption of a dominant speech by people with different ancestry. The arrival of steppe ancestry does not mean every person was replaced or every local tongue vanished immediately. Some communities may have become bilingual, some may have resisted, some may have absorbed newcomers into existing traditions. Others may have been overwhelmed. What survives in the record suggests both migration and mixture, both continuity and rupture. In Europe, the genetic impact of steppe ancestry varies by region. In some areas it becomes a major component of later populations. In others, earlier farmer ancestry remains stronger. In still others, local hunter-gatherer ancestry persists within the blend. This creates the ancestry profile that underlies many later Europeans, but it should not be confused with modern nations or simple ethnic labels. The people moving across the 3rd millennium BCE did not carry passports from the future. They lived in kin groups, clans, herding communities, and cultural networks whose names are gone. Modern attempts to claim them too neatly usually say more about modern desires than ancient realities. The transformation also had a biological dimension. Steppe-associated populations may have carried genetic variants that later became important in Europe, including some linked to lactase persistence. Though the timing and selection process were complex, they also lived with animals in ways that could affect disease exposure, diet, and mobility. Pastoralism expands human possibility, but it also ties survival to herd health, pasture access, and social negotiation over space in a dry year. Mobility can save a group in a conflict. Mobility can make a group dangerous. Ancient DNA from this period sometimes reveals stark social patterns. Certain burial sites show related males, suggesting patrilineal descent groups. Other evidence points to women moving between communities more than men in some contexts, perhaps through marriage networks. These patterns are not universal, and the evidence remains incomplete, but they hint at societies where ancestry, gender, mobility, and power were organized in ways that shaped who moved and who stayed. The genome turns social structure into a traceable pattern, but it cannot tell us how individuals felt inside it, whether a woman moved willingly into a distant household, whether a young man inherited status with pride or burden, whether a child born of mixed ancestry felt divided or simply belonged. The steppe expansion is sometimes described with the language of invasion, and in some places violence may well have played a role, but invasion alone is too blunt. The genetic and archaeological evidence points to a wider process: migration, demographic growth, social advantage, technological change, ecological opportunity, and cultural transformation unfolding over generations. No single explanation fully accounts for it. The people of the steppe did not simply ride out of the east and instantly remake Europe, yet they did remake it, profoundly enough that their ancestry, languages, and social patterns echo through later history. By the end of this transformation, Europe has become a palimpsest Ice Age foragers, Anatolian farmers, steppe pastoralists, and countless regional mixtures layered into one another. Ancient DNA exposes the drama beneath what once looked like a gradual prehistoric blur, but the story of forgotten ancestors does not belong to Europe alone. Across the ocean, in the Americas, the same science begins to disturb older certainties, revealing founding populations, lost branches, and movements through landscapes that were also transformed beyond recognition by ice, sea, and time. The Americas enter the ancient DNA story through cold, distance, and an absence of easy routes. For a long time, the standard image was simple people crossed a land bridge from Siberia into Alaska near the end of the Ice Age, followed an ice-free corridor between glaciers, and spread southward into an empty continent. It was a clean line on a map, easy to teach, easy to remember, and far too simple for the evidence that has since emerged. Ancient DNA does not erase the importance of Berenguer, but it turns the peopling of the Americas into a deeper, stranger, and more layered process than that old arrow ever allowed. Berenguer was not just a narrow bridge. During periods of lower sea level, it was an enormous region of steppe tundra connecting Northeast Asia and Northwest North America. It had animals, plants, rivers, winds, seasons, and human possibilities. People may have lived there for long periods, not merely hurried across it. Some genetic models suggest a period of isolation among the ancestors of Native Americans, before their descendants expanded through the continents. The evidence remains incomplete, partly because much of ancient Berenguer is now submerged, but the idea matters. The first Americans were not simply travellers using a temporary crossing. They may have belonged to a vanished northern world that was later drowned by the sea. Ancient DNA reveals that the ancestry of indigenous peoples of the Americas is deeply connected to ancient populations of Northeast Asia, but the branching is complex. Early lineages split, moved, and diversified rapidly once humans spread south of the ice. Some ancient genomes from North and South America show close relationships across vast distances, suggesting that people moved astonishingly fast after entering habitable regions. Archaeological evidence from sites older than the once dominant Clovis model had already challenged the idea that Clovis hunters were the first Americans. Genetic evidence reinforces the view that the story began earlier and involved populations already differentiated by the time some of the oldest sampled individuals lived. The route south remains debated. The ice-free corridor between the Great Laurentide and Cordilleran ice sheets may not have been open and ecologically usable early enough to explain all evidence for the first widespread movement. A coastal route along the Pacific has therefore become increasingly important in many models. But here, again, the sea has taken the archive. The ancient coastline people may have followed lies offshore, drowned by post-glacial sea level rise. Camps, tools, bones, and hearths that could clarify the journey may rest beneath marine sediments. Ancient DNA helps trace movement where archaeology has been partly submerged, but it cannot fully restore the lost shoreline world. A lesser-known detail is that some ancient American genomes contain signals that complicate a single founding picture. Certain studies have detected ancestry patterns in some ancient South American and Amazonian-related populations that appear distantly connected to Australasian groups, though the interpretation remains debated. This does not mean that Australians sailed directly to South America in a simple popular sense. The safer reading is that deep population structure in Asia before the peopling of the Americas may have been more complex than once thought, and that some groups entering or contributing to the Americas carried ancestry related in unexpected ways to populations far across the Pacific world. Scholars still debate how to model this signal, and it remains one of the more intriguing unresolved puzzles. Once inside the Americas, people encountered a world of extreme variety, ice margins, open grasslands, mountain corridors, deserts, forests, wetlands, coasts, and tropical lowlands all demanded different ways of living. The first people to move through these landscapes were not merely following mammoths in a single grand hunt. They adapted quickly to local ecologies, from Arctic and subarctic conditions to the high Andes and Amazonian environments. Ancient DNA, when combined with archaeology, shows both movement and regional continuity. Some populations leave descendants across long spans. Others disappear or contribute unevenly to later groups. The Americas also remind us that ancient DNA is never only scientific material. It is tied to living peoples, ancestry, sovereignty, and the treatment of the dead. Human remains in the Americas have often been studied within a painful history of colonial violence, grave disturbance, and disregard for indigenous communities. The question is not merely what a genome can reveal, but who has the right to ask, to sample, to interpret, and to rebury. In recent years, collaboration with indigenous groups has become increasingly central. Though practices vary and debates continue, the science is strongest ethically when it does not treat ancient people as ownerless data. One famous ancient genome from North America helped show a close relationship between ancient individuals and present-day Native American populations, challenging older claims that some early remains represented unrelated or mysterious outsiders. This is an important corrective. Popular speculation has often tried to detach the earliest Americans from indigenous peoples, sometimes for ideological reasons. Ancient DNA has generally strengthened, not weakened. The deep connection between ancient American remains and living indigenous communities, while also revealing complexity within that connection. The speed of early expansion is one of the great human facts in this story. Within a relatively short span, people reached far into South America, adapting to land separated by enormous distances. This movement required knowledge, flexibility, and social resilience. It also occurred in a world filled with animals now gone mammoths, mastodons, giant ground sloths, saber-toothed cats, dire wolves, ancient horses, and other megafauna. Human arrival overlaps with climatic changes and extinctions across the late Pleistocene, but no single explanation fully accounts for every disappearance. Hunting, climate stress, habitat change, and ecological disruption may all have played roles, varying by region and species. Ancient DNA from animals, sediments, and humans helps illuminate this world, but it does not reduce it to one cause. For the people living through it, the Americas were not new. They were immediate landscapes of danger and opportunity. A glacier was not a paleoclimate feature. It was a wall of ice. A coastline was not a migration corridor. It was food, storms, boats or rafts, tides, memory, and risk. A mountain pass was not a line on a map. It was breath, cold, and the decision to continue. The genetic record compresses these lives into ancestry components and branching diagrams. But behind each branch were families moving through places no human in their tradition had known before. Over thousands of years, regional histories deepen, ancient DNA reveals continuity in some areas and later population movements in others. The rise of agriculture in parts of the Americas, including maize cultivation and other domesticated crops, creates new demographic and cultural transformations. Though these histories unfold differently from the Old World, there are cities, trade networks, empires, collapses, and migrations. But the ancient genetic foundations remain tied to those first movements through Beringian and post-glacial worlds. The Americas are not a late appendix to human history. They are one of its most extraordinary expansions. And yet much remains uncertain. The earliest roots are partly drowned. Many regions remain undersampled. Preservation varies sharply by climate. Ethical concerns rightly shape what can and should be studied. Each new genome can refine the picture or disturb it. What survives in the record suggests a founding story both deeply connected to Asia and uniquely transformed by the American continents themselves. The forgotten ancestors of the Americas stand at the edge of ice and ocean, then move into everything from Arctic coasts to tropical forests. Their descendants build worlds of immense diversity. Some ancient lineages continue, others fade into mixtures, vanish from sampled records, or remain hidden because the ground has not preserved them. Like Beringia itself, part of their world is underwater, but not entirely lost. It survives in genomes, in living communities, in languages, in archaeological sites, and in the enduring fact that people entered a continent-sized unknown and made it home. From the continents, the story narrows again to places surrounded by water, where isolation magnifies ancestry, and the sea both protects and imprisons. On islands, ancient DNA reveals some of the strangest human histories of all small populations surviving at the edge of the world, voyagers crossing impossible distances, and communities that disappear so completely that only bones and blood remember them. Islands make human history sharper. On a continent, people can vanish into forests, valleys, frontiers, and neighbouring populations. On an island, the edges are visible. The sea draws a boundary around hunger, memory, marriage, and survival. It can protect a community for centuries, keeping armies, epidemics, and strangers away. It can also trap a community when crops fail, when trees are cut down, when fresh water dwindles, when storms arrive from the wrong direction, or when no canoe returns from the horizon. Ancient DNA reads islands with unusual force because isolation magnifies ancestry. Small populations drift genetically in ways large continental populations often do not. A few founders can leave a signal for thousands of years. A single later arrival can reshape the whole future. The most dramatic island stories begin with courage that later generations can barely imagine. To reach Sahel, the greater Ice Age landmass of Australia and New Guinea, early modern humans had to cross water even when sea levels were lower. Later, Austronesian-speaking voyagers moved across island Southeast Asia and into the Pacific, carrying crops, animals, pottery traditions, sailing knowledge, and social worlds across distances that look almost impossible from shore. Ancient DNA shows that these movements were not simple replacements. In places, incoming farmers and sailors mixed with older Papuan-related populations. In others, small founder groups expanded rapidly, leaving genetic signatures across vast oceanic distances. The Pacific is especially revealing. The Lapita-associated expansion carried people into remote Oceania, reaching islands no human had ever inhabited before. Their descendants eventually settled places scattered across the largest ocean on Earth. The sea was not a blank barrier to them. It was a navigable world of winds, stars, currents, birds, clouds, and remembered routes. Yet DNA shows that even masterful voyaging did not erase older layers. In many Pacific populations, ancestry reflects mixtures between Asian-related Austronesian ancestors and Papuan-related groups, with timing and proportions varying by island and region. Scholars still debate the exact sequence of contacts, marriages, and return voyages, but the genetic record makes one thing clear: the Pacific was never empty space between islands. It was a connected human world. Island isolation can also preserve strange survivals. On some islands, Ancient DNA reveals continuity across long periods despite outside contact. On others, it reveals sudden transformation. A small incoming group may bring new ancestry, language, crops, or status systems, while older people are absorbed or pushed aside. Because island populations can be small, chance matters more. A storm can kill a lineage. A canoe carrying a few families can alter the ancestry of an entire island. A taboo. Marriage rule, or chiefly system, can shape who has descendants centuries later. The genome records these outcomes, though it cannot fully explain the choices behind them. A lesser-known island lesson comes from the Caribbean. Ancient DNA has shown that the population history of the Caribbean before European conquest was more complex than older models suggested, involving movements from South America and interactions among island communities over time. Some earlier populations were largely replaced or absorbed by later ceramic age groups, but not in a single, simple sweep. Then, European colonization brought catastrophe on a scale that shattered the demographic world. Violence, enslavement, forced labour, disease, and displacement devastated indigenous communities. Yet ancient and modern DNA also show indigenous Caribbean ancestry surviving in living people, even where colonial narratives once claimed total disappearance. The word extinct, when applied to human communities, can hide survival in the bodies of descendants. Islands also expose the danger of telling collapse stories too neatly. Rapa Nui, often called Easter Island, has long been used as a moral tale about ecological self-destruction. The popular version imagines a society cutting down all its trees, collapsing into chaos, and standing as a warning to the modern world. Archaeology and genetics have complicated that story. Environmental change and deforestation were real, but the timing, scale, and social consequences remain debated. European contact brought disease, slaving raids, and colonial violence that profoundly damaged the population. Ancient DNA has helped test claims of prehistoric contact and population continuity, but no single explanation fully accounts for the island's history. The simpler the collapse story sounds, the more carefully it must be handled. The same caution applies elsewhere. Islands can produce dramatic ruins and lonely statues, but human history is rarely a fable with one villain. Climate shifts, resource pressure, social hierarchy, outside contact, disease, warfare, trade dependence, and chance can all converge. Ancient DNA does not remove the drama. It prevents the drama from becoming too easy. In the North Atlantic, islands tell a different kind of story. The settlement of Iceland, for example, can be traced genetically to Norse and Gaelic sources, reflecting the movements of Viking Age people, including women from the British Isles and Ireland. There, ancient and modern DNA reveal migration, social inequality, and the human consequences of expansion across cold seas. The sagas preserve one kind of memory, genes preserve another. Neither is complete alone. But together they show how an island society can be founded by people whose origins were already mixed through raiding, settlement, marriage, and enslavement. Some islands hold even older mysteries: the Andaman Islands, New Guinea Highlands, Madagascar, Sardinia, the Canary Islands, and countless smaller places each preserve distinctive histories of isolation and connection. Madagascar, settled by people with ancestry from both Southeast Asia and East Africa, is a remarkable example of long-distance movement across the Indian Ocean. Sardinia preserves unusual amounts of ancestry related to early European farmers, partly because later steppe-related ancestry affected it less than many mainland regions. The Canary Islands held indigenous populations with North African connections before European conquest transformed their world. In each case, ancient DNA does not flatten identity into percentages. It reveals how islands can become vaults of older ancestry and crossroads of astonishing journeys at the same time. But isolation has a cost. Small island populations may pass through bottlenecks, where only a few individuals contribute to later generations. This can amplify rare genetic variants, reduce diversity, and make communities vulnerable to disease or environmental shock. The same sea that protects can limit rescue. If a drought strikes a continental valley, people may move. If a harvest fails on a remote island, escape may require boats, weather, and knowledge that are not always available. Ancient DNA can detect bottlenecks, founder effects, and sudden ancestry shifts, but the human experience behind them would have been fearfully immediate empty storage pits, dying crops, arguments over food, prayers to ancestors, and eyes searching the horizon for sails. The most powerful island histories are therefore not only about disappearance, they are about endurance under constraint. People on islands learn the details of their worlds with extreme precision where fresh water hides, which reefs are dangerous, when birds nest, how storms announce themselves, which trees are strong enough for canoes, which marriages keep peace, which stories preserve roots. Their survival depends on knowledge braided through generations. When those societies vanish or transform, the loss is not only genetic, it is ecological memory, navigation, ritual, language, and an entire way of belonging to a bounded world. Ancient DNA can sometimes rescue a fragment of that loss. It can show that a supposedly vanished people contributed to later populations. It can reveal that an island was settled earlier than expected, or by people from an unexpected direction. It can prove that isolation was not absolute, that canoes returned, that strangers became kin. It can also show abrupt breaks, where a population leaves little trace after conquest, epidemic, or replacement. The evidence remains incomplete. Especially in warm island climates where DNA decays quickly, but every successful genome is like a lamp lit briefly in a room almost swallowed by dark. By the time the island stories are gathered together, they reveal a paradox at the heart of ancient DNA. The smallest places can contain the widest histories. A remote island cemetery may hold evidence of transoceanic navigation, colonization, disease, survival, and erasure. A single skeleton can connect Asia, Africa, Europe, Oceania, or the Americas in ways no local legend alone could preserve. The sea separates, but it also carries, it hides, but it also remembers. And from islands, the story turns inward again, back into the body itself. For ancient DNA is not found only in grand migrations or vanished peoples. It is sealed in teeth, bone, and the microscopic traces of ordinary lives. Inside those remains are clues not only to ancestry, but to hunger, family, illness, violence, and the intimate details of people who never expected the future to read them. A tooth is one of the body's smallest archives, and one of its most faithful. Long after skin is gone, after hair has vanished, after the soft organs have returned to soil, teeth remain with a stubborn mineral patience. They survive fire better than flesh, time better than bone, and handling better than many other traces of the dead. To an earlier archaeology, a tooth could reveal diet, age, wear, and sometimes disease. To ancient DNA, it becomes something more intimate. It is a sealed chamber. A hard white container where ancestry, childhood, infection, and kinship can remain protected after everything else has been stripped away. The reason teeth matter so much is partly anatomical. Inside the tooth, especially within dentine and the pulp chamber, genetic material can be sheltered from some of the damage that destroys DNA elsewhere. The hard enamel on the outside is not rich in DNA, but it protects what lies beneath. A molar from a burial, a loose tooth from a cave floor, or a jaw fragment from a forgotten cemetery can therefore carry information about a person's family, population, and sometimes the pathogens that move through their bloodstream before death. It is difficult to imagine a more private record. The mouth that once chewed, spoke, laughed, cried out, or went silent in illness becomes a vessel for evidence thousands of years later. Ancient DNA from teeth and bones has changed how archaeologists understand graves. A burial is no longer only a pattern of objects and posture. It can become a family map. People buried side by side may turn out to be parent and child, siblings, cousins, or unrelated individuals joined by social bonds rather than blood. A richly furnished grave may belong to someone genetically local, or to a person whose ancestry points far away. A child buried with care may reveal that status was inherited. Not earned, a mass grave may show whether the dead were one community, a captured group, victims of disease, or members of a family line destroyed together. These are not small details. They change the emotional meaning of archaeological sites. In some prehistoric graves, ancient DNA has revealed close kinship across generations, showing that tombs were reused by families who remembered or maintained rights to the dead. In others, it has shown surprising lack of biological relationship, suggesting that burial communities could be social, ritual, or political rather than simply familial. The evidence remains incomplete because not everybody preserves DNA, and not every burial is sampled. But the pattern already warns against easy assumptions. A graveyard is not always a family cemetery. A shared tomb is not always shared blood. A person can belong by ancestry, marriage, adoption, rank, captivity, ritual, or need. Teeth also preserve the violence of history in ways both direct and subtle. The genome can identify sex where bones are damaged or ambiguous. It can show whether victims in a massacre included local people or outsiders. It can reveal that men, women, and children died together, or that one group was targeted more than another. Combined with trauma on bones, isotope evidence from diet and movement, and the positions of bodies, ancient DNA helps reconstruct events that no witness recorded. Sometimes the result is grimly clear: a community attacked, a family line ended, bodies thrown into pits. Sometimes the result remains uncertain. A violent death can be murder, warfare, execution, sacrifice, or social punishment. No single genetic test can explain motive. One lesser-known surprise is that ancient DNA can survive in dental calculus, the hardened plaque that clings to teeth. To most people, calculus is an unpleasant trace of poor dental cleaning. To archaeologists, it is a mineralized time capsule. It can trap DNA from oral bacteria, food particles, plant remains, proteins, and microscopic debris from daily life. This means the mouths of the dead can preserve evidence of diet, health, and environment in astonishing detail, traces of milk proteins. Plant starches, fibers, and microbial communities can survive where ordinary organic remains have disappeared. The plaque that once irritated a living mouth becomes a record of meals, habits, and disease. This is where ancient DNA widens beyond ancestry. It begins to show life as lived in the body. The transition to farming, for example, did not only change settlement patterns, it changed mouths. Diets richer in carbohydrates affected oral bacteria and dental disease. Living with animals changed exposure to pathogens. Denser settlements allowed infections to circulate more easily. The body absorbed civilization before civilization wrote its own history. Cavities, abscesses, bone lesions, and microbial DNA all become evidence that the great transformations of human society were also biological events. Pathogens are especially revealing. Sometimes the DNA recovered from human remains is not only human, it belongs to bacteria, viruses, or parasites that infected the person before death. A tooth can preserve the genetic signature of plague, tuberculosis, leprosy, hepatitis, or other diseases, depending on preservation and sampling. This turns cemeteries into epidemiological archives. It allows researchers to track how diseases evolved, how they spread, and whether ancient outbreaks were caused by pathogens related to later historic pandemics. The dead do not only reveal who they were, they reveal what lived inside them, yet the human scale must not be lost. A pathogen genome can identify an ancient infection, but it cannot show the face of fever. A child's DNA can reveal kinship, but not the sound of mourning. A skeleton can show nutritional stress, but not whether hunger came from failed crops, unequal distribution, exile, drought, or war. Scholars still debate many interpretations because biological evidence rarely speaks alone. The body records consequences. The causes must be reconstructed carefully from context. Ancient DNA also exposes movement within ordinary lives. Isotopes in teeth can show where a person spent childhood, while DNA can show ancestry, and burial context can show where they died. Together, these methods reveal migrants, outsiders, marriage partners, captives, traders, or children moved between communities. In some ancient societies, women appear more likely than men to have moved between groups, perhaps through marriage practices. In others, mobility patterns differ. What survives in the record suggests that kinship and residence were organised in many ways, not according to one universal prehistoric rule. The effect is to make ancient communities feel less abstract. A village is not just a dot on a map. It is a web of related and unrelated people, some born nearby, some arriving from elsewhere, some carrying childhood diets different from their neighbours, some buried with objects that match local customs despite foreign ancestry. Identity becomes layered. A person may be genetically distant but socially central. Another may be local by ancestry but buried without honour. Ancient DNA helps reveal these tensions, but it also reminds us that biology is not destiny. People have always made belonging out of more than blood. There is a quiet irony here. The body parts that survive best are often the parts least associated with personality. A tooth, a femur, a petrous bone from the skull-hard fragments, silent and anonymous. Yet these fragments can restore the outlines of lives that monuments ignore. Kings and conquerors had inscriptions. Ordinary people had bones. Ancient DNA gives those bones a kind of testimony, not poetic, not complete, but powerful enough to complicate the story of whole societies. The secrets buried in teeth do not always lead to grand migrations or vanished human species. Sometimes they reveal a mother and child, a person who grew up far from where they were buried, a pathogen that entered a town, a diet that changed with farming, a community where relatives lay together for generations. These small findings matter because deep history is not only made by continental movements, it is made by bodies under pressure, by families forming and breaking, by diseases passing invisibly, by hunger written into enamel, by violence leaving marks that survive longer than names. And once ancient DNA begins to recover the microbes of the past, the human story darkens in a new way. The same methods that reveal ancestry also reveal ancient enemies that followed people into farms, towns, armies, ships, and cities. Some of these enemies changed history as surely as migrations did. They did not build monuments, speak languages, or leave tools. They moved through breath, blood, fleas, water, and touch. They were plagues, and their DNA waits in graves. The grave does not always say plague at first. It may look like any other burial ground disturbed by time bones laid in rows, bodies placed with care, a cemetery used during a difficult season, a pit filled more quickly than usual. The dead themselves may show no obvious mark of what killed them. Many infectious diseases leave little on the skeleton. Fever does not carve its name into bone. A person can die in agony and leave behind a body that, centuries later, appears almost peaceful. For a long time, this made ancient epidemics difficult to prove. Historians had texts for some outbreaks. Archaeology had mass graves for others, but the invisible killers remained uncertain. Ancient DNA changed that silence. In the pulp of teeth, in bones, and sometimes in preserved tissues, researchers began finding the genetic remains of pathogens. The dead were not only carrying their own ancestry into the future, they were carrying the ancestry of the microbes that killed them. This opened a new history beneath history, the evolution of disease itself: plague, tuberculosis, leprosy, smallpox-related viruses, hepatitis, malaria. And other infections could now be studied not only as written disasters, but as organisms with lineages, mutations, expansions, and disappearances. The most famous of these ancient enemies is plague, caused by the bacterium Yersinia pestis. It is tied in the public imagination to the Black Death of the 14th century, when enormous numbers of people across Europe, the Middle East, and North Africa died. But ancient DNA has revealed that plague is far older than medieval catastrophe. Strains of Yersinia pestis have been detected in prehistoric human remains from Eurasia thousands of years before the Black Death. These early forms were not always identical to later bubonic plague strains, and scholars still debate how they spread and how deadly they were. Some lacked genetic features associated with efficient flea-borne transmission. Even so, Their presence means that plague-like disease was moving among ancient populations long before cities of the Middle Ages became mass graves. This discovery changes the meaning of prehistoric population shifts. When archaeologists see settlements abandoned, populations declining, or genetic turnovers occurring, climate, migration, warfare, and economic change are often considered first. Disease is harder to see. Ancient pathogen DNA gives it a place at the table. It does not mean every collapse was caused by plague. That would be too simple, but it shows that disease may have been one pressure among many. Especially as people became more mobile, more connected, and more dependent on animals and dense communities. The rise of farming helped to create new disease landscapes. Settled villages concentrated people, waste, stored food, rodents, and domesticated animals. Herds brought humans into closer contact with animal pathogens. Grain storage attracted pests. Larger populations allowed infections to sustain themselves where small scattered bands might have broken transmission chains. Civilization, in its earliest forms, did not merely build houses and fields, it built opportunities for microbes. This is one of the grim bargains of human success. The same social density that supports growth can also support contagion. Ancient DNA allows researchers to follow that bargain across time. The bacterium that causes leprosy has been recovered from ancient remains, showing how strains spread and changed. Tuberculosis has deep roots and a complicated history involving humans and animals. Hepatitis B has been found in ancient genomes, revealing infections in people who lived thousands of years ago. These findings are still developing, and the evidence remains incomplete. Because pathogens preserve unevenly, and not every infection enters the bloodstream or survives in sampled tissues. But each recovered genome turns disease from a vague historical force into an evolutionary actor. A lesser-known detail is that pathogen DNA can sometimes appear in individuals who were not buried in obvious epidemic contexts. A single skeleton in an ordinary grave may carry evidence of infection, suggesting that ancient disease history is not only written in mass death. Chronic illnesses, local outbreaks, hidden carriers, and infections that killed quietly all leave scattered traces. The dramatic plague pit matters. But so does the lone tooth from a person whose community may never have known that a microbe inside them would one day become part of a global history of disease. The Justinianic plague, beginning in the 6th century, is one example where ancient DNA has clarified long-standing debates. Historical sources describe devastating outbreaks in the Byzantine world and beyond, but scholars have debated their scale, impact, and role in political and economic change. DNA from early medieval remains has confirmed that Yersinia pestis was involved in at least some outbreaks associated with this pandemic. Yet confirmation of the pathogen does not settle every question. How many people died in each region, how societies recovered, and how much the plague contributed to broader transformations remain debated. The microbe is real. The consequences still require careful interpretation. The Black Death is clearer in some ways and still mysterious in others. Ancient DNA confirms Yersinia pestis in many plague burials, and genetic studies show how strains relate to later outbreaks. The disease moved through trade routes, towns, ports, and countryside with terrifying speed. But even here, the story is not only biological. Climate, rodent ecologies, flea vectors, trade networks, war, famine, urban crowding, and social responses all shaped the pandemic. A bacterium does not travel through history alone. It moves through human systems. That is the larger lesson. Ancient pathogen, DNA does not replace social history. It gives social history a dangerous living partner. A plague strain may emerge, but roads, ships, markets, armies, migration, and inequality determine where it goes and whom it reaches. Disease exposes the hidden architecture of human connection. The same exchange networks that carry amber, obsidian, grain, silk, or silver can carry death. A port city is a gateway for wealth and infection. A pilgrimage route can bind believers and spread fever. An army can move borders and microbes at the same time. There is also a question of survival. Ancient DNA from pathogens can sometimes show that a disease changed over time, but human populations change too. Immune-related genes may shift under selective pressure from infections. Some variants inherited from archaic humans may have affected immune responses. Later historical epidemics may have shaped the genetic landscape of survivors. These possibilities are difficult to prove in simple ways, and scholars still debate many proposed links. But the principle is clear. Disease is not only something that happens to history, it helps shape the bodies that continue after catastrophe. The emotional weight of this is considerable. A cemetery from an epidemic is not just evidence of mortality, it is evidence of interruption, harvests left unfinished, children losing parents, artisans gone from workshops, houses closed, rituals compressed because too many have died too quickly. Ancient DNA identifies the pathogen, but the human scene must be imagined from the surrounding traces: hurried burials, unusual pits, missing grave goods, or, sometimes, careful rites maintained even under pressure. People facing epidemic death often tried to preserve order when order itself was failing. No single explanation fully accounts for how plagues transform societies. Sometimes populations rebound. Sometimes labour shortages shift power. Sometimes states weaken. Sometimes religious movements intensify. Sometimes scapegoating and violence follow. Sometimes memory turns epidemic into legend. The biological event becomes social, political, and spiritual. Ancient DNA can prove that a pathogen was present, but it cannot measure fear, blame, prayer, or grief. And yet, by recovering ancient disease, it brings the past closer in an unsettling way. The people of deep time were not only distant ancestors moving through grand migrations, they were bodies vulnerable to invisible life. They coughed, weakened, fevered, carried infections to those they loved, and sometimes survived because of chance, immunity, care, or isolation. Their forgotten ancestors were not only human, some were microbial lineages that travelled with them, fed on them, and shaped their descendants. As ancient DNA follows these buried pathogens, it also forces a return to the deepest homeland of humanity, where the story is older than Neanderthals, older than Denisovans, older than farming, steppe migrations, and the peopling of the Americas. Africa holds the longest human history and the greatest human diversity, but its ancient DNA has been hardest to recover. In that difficulty lies another mystery. The continent most central to who we are still contains some of the most hidden genetic chapters of all. Africa is not the beginning of the human story in a simple ceremonial sense. It is the deep ground of it, long before farming villages, steppe migrations, island voyages, or the first crossings into the Americas. Africa holds the long and complicated making of Homo sapiens. It is where the oldest branches of our species take shape, where bodies, minds, languages, tools, and social worlds develop across landscapes of astonishing variety. Desert, savanna, rainforest, coast, highland, wetland, and river corridor all shape human possibility, yet ancient DNA, the very tool that has illuminated so much of Eurasia's past, struggles here more than almost anywhere. The reason is brutally ordinary heat and moisture destroy DNA. In many African environments, ancient genetic material decays quickly. Bones that might preserve DNA for tens of thousands of years in Siberian cold may yield little or nothing in warmer conditions. This creates a cruel imbalance. The continent most essential to human origins is the continent where ancient DNA often survives least well. The result is not absence of history, but absence of preservation. Africa's past is not silent because it was simple. It is silent in places because chemistry has burned the pages. Even so, what has emerged is transformative. Ancient and modern DNA show that African populations contain the deepest human genetic diversity on Earth. This diversity reflects long histories of separation, mixture, adaptation, and movement within the continent. Human origins were not the story of one small group in one neat location suddenly becoming modern and walking outward. The evidence points instead toward a more structured process, with populations across parts of Africa connected and separated over long periods. Scholars still debate the geography and timing of these processes, but the old image of a single cradle is giving way to something wider and more dynamic, a braided African origin made across regions and climates. Some of the most ancient divergences among living human populations are found within Africa. Groups in southern Africa, Central Africa, Eastern Africa, and elsewhere preserve lineages that split from one another very deeply in time. Though no living group is a frozen relic of the past, this point matters. Ancient ancestry does not make a people unchanged or primitive. Every living population has continued evolving, adapting, mixing, and making history. The deep lineages inside African DNA reveal not living fossils, but the immense depth of African human history. A lesser-known surprise is that Africa may also preserve traces of archaic admixture from populations that are not yet known from fossils in any clear way. Some genetic studies have suggested that ancestors of modern Africans may have mixed with deeply divergent human groups within Africa, sometimes described as ghost archaic populations. These were not Neanderthals or Denisovans in the familiar Eurasian sense, but possibly other lineages that separated from the ancestors of modern humans long before later mixture occurred. The evidence remains debated, partly because modelling deep ancestry without ancient genomes is difficult, but the possibility reminds us that Africa, too, may once have contained multiple forms or deeply structured populations of humans whose bones have not yet been securely connected to genetic signals. The geography of Africa makes this plausible. The Sahara, now often imagined as a barrier, has repeatedly changed. At times, it was far greener, crossed by rivers, lakes, grasslands, animals, and people. At other times, it became a vast dry expanse that separated populations and redirected movement along coasts, mountains, and river systems, the Nile Corridor, the Red Sea region, the Horn of Africa, the Sahel, the Congo Basin, southern African landscapes. And coastal zones all offered different possibilities. Climate did not merely challenge human groups, it rearranged their relationships. A green Sahara could connect worlds, a dry Sahara could divide them. Over tens of thousands of years, those cycles helped shape genetic structure and cultural exchange. Ancient DNA recovered from African remains has begun to reveal movements that were once nearly invisible. Pastoralism spread through parts of the continent, bringing herding lifeways into regions where hunting, gathering, fishing, and foraging had long histories. Later, the expansion of Bantu-speaking populations transformed much of sub-Saharan Africa, carrying farming, ironworking in some contexts, languages, and ancestry across enormous distances. Ancient DNA helps show that these movements involved mixture with local foragers and pastoralists, not simple replacement everywhere. The people who made later African societies were layered, like those elsewhere, though the layers are often harder to sample. The spread of herding into Eastern Africa is especially revealing. Genetic evidence shows interactions among groups connected to Northeast Africa, Eastern African foragers, and later populations. Cattle, sheep, and goats were not only economic resources, and they transformed mobility, diet, status, marriage, and relations with landscape. In some places, pastoralism flourished in environments where farming was difficult. In others, herders interacted with farmers and foragers in complex ways. What survives in the record suggests that African prehistory was full of shifting lifeways, not a static background waiting for later historical kingdoms. Africa also resists the temptation to equate ancestry with language or culture too neatly. The continent contains extraordinary linguistic diversity, and language shifts can occur through migration, elite influence, trade, intermarriage, or social transformation. Genetic ancestry may spread with language in some cases and not in others. A community may adopt a language without major genetic change, or migrants may be absorbed while their speech persists. This is true everywhere. But in Africa's deep and recent history, it is especially important. Ancient DNA can illuminate movement, but it cannot hear the lost languages that once filled the air. The consequences of undersampling Africa are significant. Because more ancient DNA has been recovered from Europe and northern Eurasia, global narratives can become distorted, as if the best sampled regions were the most important. They are not. They are often simply colder, more excavated, and more heavily funded. The human story cannot be centered only where DNA survives easily. If anything, the gaps in African ancient DNA should make us more cautious about all human origin stories. The missing evidence may not be minor. It may contain entire chapters. Still, every new African genome matters profoundly. It can reveal population continuity where disruption was assumed. It can expose movement where isolation was imagined. It can show that ancient foragers contributed to later peoples. Even when their lifeways disappeared, it can clarify how pastoralists, farmers, and hunter-gatherers interacted over thousands of years. It can also challenge modern categories imposed by colonial history, nationalism, or simplistic racial thinking. Africa's genetic past is not reducible to a few labels. It is the deepest reservoir of human variation. This is where ancient DNA becomes not only a scientific tool, but a corrective to old mistakes. For centuries, many outside narratives treated Africa as lacking history, before written records or external contact. Archaeology had already proven that false. Ancient DNA makes it even more untenable. The continent's past is vast, active, and foundational. It contains lost populations, climate transformations, technological innovations, migrations, survivals, and mixtures as dramatic as any elsewhere. The difference is that many of its archives are harder to open. The forgotten African lineages are not marginal to humanity. They are central. Some are ancestral to everyone alive. Others contributed to particular regions or vanished as distinct groups. Some may remain only as faint signals in living genomes, awaiting ancient remains that may never preserve enough DNA to confirm them. No single explanation fully accounts for the depth and diversity of African ancestry, because the story is older than the tools now used to study it. As ancient DNA pushes further into Africa, it also forces a larger question. If ancestry everywhere is layered, mixed, and changing, what happens to the old ideas people use to define themselves? If every population is a product of movement and contact, if vanished ancestors live inside living bodies, if identity has never been biologically pure, then ancient DNA does more than rewrite prehistory. It unsettles the stories modern people tell about race, belonging, civilization, and inheritance. Ancient DNA does not simply add new facts to the human story. It changes the shape of the story people thought they were living inside. For a long time, ancestry was often imagined as a line, a people rising from a homeland, remaining itself through time, carrying an essence forward from ancestors to descendants. Nations, tribes, empires, and families have all told versions of this story. Some are poetic, some political, some sacred, some dangerous. They give people belonging. But they can also harden into myths of purity, priority, and exclusion. Then the ancient genomes arrive, and the line becomes a braid. Everywhere the evidence is strong enough to see clearly, the same pattern returns. Populations move, they mix, they split, they absorb strangers, they disappear as separate groups while surviving inside others. Europe is not one ancient people, but repeated layers of hunter-gatherers, farmers, steppe pastoralists, and later movements. The Americas are not a simple late arrival story, but a deep branching history connected to Asia and transformed by the continents themselves. The Pacific is not isolation alone, but voyaging, mixture, and founder effects across the ocean. Africa is not a static origin point, but the deepest and most complex field of human diversity. Even the boundary between modern humans and other human forms turns out to be porous. Neanderthals and Denisovans are gone, yet pieces of them remain. This does not mean identity is meaningless. It means identity is not the same thing as genetic purity. Human groups are real historically, culturally, linguistically, politically, and emotionally, but they are not sealed biological containers. A people can maintain language, ritual, law, memory, and belonging while its ancestry changes. Another group can share ancestry but become culturally different through migration, conquest, adoption, or time. Ancient DNA makes it harder to pretend that blood and culture move together in perfect lockstep. They never have. This is one of the most important corrections the science offers. Race, as commonly imagined in modern societies, does not map neatly onto the deep structure of human genetic variation. Human genetic differences exist, and ancestry can often be studied with great precision, but the old racial categories are blunt, recent, and politically loaded. They slice across gradients, mixtures, and histories far older than the categories themselves. Ancient DNA does not prove that humans are all identical. It proves something more interesting: humans are different in patterns shaped by movement, isolation, adaptation, and mixture, and those patterns do not support simple myths of pure races. A lesser-known consequence is that ancient DNA can unsettle nationalist archaeology. In many parts of the world, prehistoric peoples have been claimed as direct ancestors of modern nations, sometimes with great confidence and little evidence. A burial mound, a pottery style, or a skeleton becomes a political symbol. Ancient DNA can complicate those claims by showing migrations and mixtures that do not fit modern borders. It can reveal that the people buried under a modern country's soil may be genetically closer to groups elsewhere, or that today's population descends from several ancient sources rather than one heroic origin. This can be uncomfortable, especially where identity has been built on deep continuity. The evidence does not erase belonging to place, but it does challenge simple possession of the past. At the same time, the science can be misused. Genetic ancestry results can be flattened into slogans. Percentages can be treated as destiny. Ancient populations can be recruited into modern ideologies they would not recognize. Steppe ancestry, hunter-gatherer ancestry, farmer ancestry, Neanderthal ancestry, or any other category can become a badge in arguments about superiority or authenticity. This is a distortion. The categories are tools for studying history, not ranks of human worth. The people they describe were not symbols. They were living communities with their own fears, skills, and contradictions. The ethical questions are especially sharp because ancient DNA is taken from human remains. A bone is not just data. It belonged to a person. In some cases, that person may have living descendants, cultural heirs, or communities with strong beliefs about burial, disturbance, and ancestry. The power to extract DNA from the dead must be matched by responsibility. Researchers increasingly work with descendant communities, indigenous groups, local scholars, and legal frameworks. Though the practice is uneven and debates continue, the evidence remains incomplete not only because preservation is difficult, but because not every question should be answered in the same way, and not every grave should be treated as a resource. Ancient DNA also changes how we think about civilization. Older histories often describe civilization as the achievement of distinct peoples moving upward through stages, hunter-gatherers to farmers, farmers to cities, cities to states. The genetic record cuts across that ladder. It shows that the makers of civilizations were themselves products of earlier mixtures, migrations, collapses, and absorptions. The first farmers were not timeless farmers. Steppe pastoralists were not untouched horse lords. Islanders were not isolated until outsiders arrived. Africans were not a single ancestral source. Every society stands on deeper human movement. This makes the past less tidy, but more humane. The rise of a civilization no longer appears as the natural flowering of one pure lineage. It becomes the work of people inheriting many histories at once. A city wall may be built by descendants of migrants and locals. A language may be spoken by people whose ancestry comes from several directions. A royal tomb may contain a ruler whose genetic background complicates later claims of dynasty and origin. The grandeur remains, but the foundation becomes more honest. There is also humility in discovering how much of humanity comes from disappearance. Many ancestors did not leave monuments, texts, or named cultures. Some survive only as tiny segments of DNA. Others are inferred from statistical ghosts. Others are gone even from genetics, because their lineages ended entirely. The living are not the inevitable summit of everyone who came before. They are the descendants of those whose lines happen to continue. Behind every surviving lineage lie countless broken ones. No single explanation fully accounts for why some populations endure and others vanish. Numbers matter, climate matters, disease matters, technology matters, violence, marriage, geography, luck, and social structure all matter. Ancient DNA can show outcomes with startling clarity, but the reasons remain layered. A vanished population may have been conquered, absorbed, starved, scattered, or transformed so gradually that no one living through it experienced disappearance as a single event. Identity can end without a final battle. A people can dissolve through marriage, language shift, adoption, migration, or the quiet fact that grandchildren belong to a different world. This is why ancient DNA is both intimate and destabilizing. It tells modern humans that they are carrying the forgotten dead, but not in the simple way family legends often promise. The ancestors inside us may include strangers, enemies, refugees, captives, neighbours, extinct relatives, and people whose names would have meant nothing to one another. Each person is not a pure vessel from one past, but a meeting place of many pasts. The body becomes an archive of encounters. And perhaps that is the deeper revelation. Human identity has always been made at borders between species, between climates, between lifeways, between islands and continents, between the living and the dead. Ancient DNA does not reduce humanity to biology. It shows biology recording the consequences of human life movement: love, coercion, survival, hunger, adaptation, and loss. The genome is not the soul of a people, but it is one of the traces a people leaves behind when everything else is gone. By the time this truth settles in, the forgotten ancestors no longer seem remote. They are not only skeletons in caves, farmers under long barrows, steppe herders beneath mounds, islanders in wind-cut cemeteries, or children buried during plague. They are part of the living fabric, present and absent at once, and the science that reveals them is still young. The next discoveries may come from sediments, proteins, new sequencing methods, or bones already sitting unnoticed in museum collections. The past has not finished changing, because the tools used to read it are still changing too. The future of ancient DNA may begin with something that does not look like a discovery at all. A box in a museum storeroom, a cave sediment sample sealed decades ago, a tooth too damaged for older methods, a bone fragment once labelled animal, a burial excavated before anyone knew that the dead still carried genetic archives inside them. The great revelations may not all come from new expeditions into remote landscapes. Some are already waiting in drawers, mislabelled, overlooked, or considered too small to matter. Ancient DNA has taught researchers to respect fragments. A fingertip can reveal Denisovans. A tooth can reveal plague. A pinch of cave soil can reveal humans who left no bones at all. The technology keeps growing more sensitive. Earlier researchers needed relatively well-preserved remains, often from cold environments. Now, improved methods can recover shorter, more damaged fragments and distinguish ancient DNA from modern contamination more effectively. Scientists can study not only human genomes, but also DNA from animals, plants, microbes, and sediments, building entire vanished environments around the people who lived in them. A cave layer may reveal Neanderthals, hyenas, deer, plants, changing temperatures, and the arrival or disappearance of human groups. The dead are no longer isolated specimens. They are part of ancient ecosystems, becoming visible molecule by molecule. There is also growing work beyond DNA itself. Proteins can survive where DNA fails, sometimes allowing researchers to identify species or relationships from fossils too old or too damaged for genetic sequencing. This matters enormously in warm regions where DNA preservation is poor. It may help open older African and Asian chapters that remain hidden. The evidence remains incomplete, but the direction is clear. The future of human origins will not depend on one kind of trace. It will be built from DNA, proteins, isotopes, sediments, fossils, tools, climate records, and living communities whose histories cannot be reduced to laboratory data. Yet the power of the science makes its ethical questions sharper. Each new method can extract more from smaller remains, but that does not mean every extraction should happen. Ancient people were not raw material waiting for modern curiosity. They belonged to communities, landscapes, and sometimes to descendants who still care deeply about their treatment. The future of ancient DNA will depend not only on cleaner labs and stronger computers, but on trust, consent, collaboration, and humility. Without those, the science risks repeating older forms of taking from the dead and from the living. The discoveries still ahead may be astonishing. More ghost populations may gain clearer outlines. Denisovans may finally acquire faces through better fossils. Africa's hidden lineages may become less hidden. The drowned coastlines of early migration may be approached through underwater archaeology and environmental DNA. Ancient disease histories may reveal why some pandemics burned through societies while others faded. But no final genome will complete the human story. Each answer will open another absence. What ancient DNA ultimately reveals is not a single forgotten ancestor, but a world crowded with them. Some are close, some remote, some human in familiar ways, some archaic and strange, some known only through statistical echoes. They remind us that humanity was never one clean thread. It was always crossings, losses, survivals, and returns. The past is not fixed behind us. It is still being read. The last image is not a laboratory, though the laboratory is where so much of this hidden past becomes visible. It is not a chart of ancestry, though those branching lines have changed what can be known. It is a landscape at dusk, somewhere no longer exactly as it was a drowned plain beneath the North Sea, a Siberian cave mouth darkening in cold air, a green Sahara lake bed now dry beneath stars, a Pacific island ridge above a restless ocean, a valley where farmers once cut the first fields into older hunting grounds. Across those places, people moved without knowing they were becoming ancient. They carried children, tools, seeds, stories, infections, fears, and fragments of ancestors already forgotten. Some watched ice retreat, some watched forests rise, some crossed water toward islands no human had seen before, some buried their dead with care, never imagining that thousands of years later a tooth, a bone, or a trace in the soil would speak for them. Ancient DNA does not bring them back whole. It cannot restore their voices or return their names, but it gives the silence texture. It shows that disappearance is not always the end, that vanished people can remain in the living, that a forgotten meeting beside an ice age river can still echo inside a body today. It also shows how fragile every world is. Populations that endure for millennia can fade. Landscapes that seem permanent can drown, dry, freeze, or bloom. Civilizations rise later on foundations made from countless lives, no monument records. In the end, the genome is not a simple answer to who we are. It is a reminder that who we are has always been shared with the missing. We are not separated from deep time by a wall, but connected to it by thin, broken, persistent threads. Some pass through caves, some through graves, some through disease, some through love, violence, migration and survival, and some pass quietly through us. The forgotten ancestors remain mostly beyond reach, but not entirely. They wait in ice, in dust, in teeth, in sediments, in the bodies of descendants, and in the changing earth itself. The deeper scientists look, the less lonely the human story becomes. Behind every living person stands a crowd of the unnamed, and behind them, more shadows, stretching back into landscapes that have vanished under forest, desert, glacier, and sea. The past is still there, not as a place we can return to, but as a trace we carry forward, breathing with it, walking with it, and leaving our own fragile record for whatever future learns how to read us.